Welcome to our microbiology classes. Today I discuss about general microbiology, that is bacteriology. What is microbiology? Microbiology is the study of microorganisms, a large and diverse group of microscopic organisms that exist as single cell or cell cluster. It also includes viruses, which are microscopic but not cellular. There are two types, eukaryotes and prokaryotes. Biologic relationship of pathogenic microorganisms. Animal pathogenic microorganism helminth. Type of cells eukaryotic. Plant type of cell eukaryotic. Protist protozoa and fungi. Type of cell eukaryotic. Prokaryote pathogenic microorganism bacteria, that is prokaryotic. Viruses non-cellular. Important features: cells have a nucleus or nucleus containing DNA, surrounded by cytoplasm, within which proteins are synthesized and energy is generated. What is virus? Virus have an inner core of genetic material, either DNA or RNA, but no cytoplasm. Method of replication: either by binary fission or by mitosis. Nature of the nucleic acid. Cells contain both DNA and RNA. Viruses contain either DNA or RNA, but not both. Eukaryotic cell has a true nucleus with multiple chromosomes, surrounded by nuclear membrane, and uses a mitotic apparatus to ensure equal allocation of the chromosome to progeny cells. The nucleus of the prokaryotic cell consists of a single circular molecule of loosely organized DNA lacking a nuclear membrane and mitotic apparatus. Prokaryotic bacterial cells versus eukaryotic human cell. DNA within a nuclear membrane is present in eukaryotic cell. DNA associated with histone is present in eukaryotic cell. Mitotic division present in eukaryotic cell. Chromosome number 1 in prokaryotic cell and more than 1 in eukaryotic cell. Membrane bound organelles present in eukaryotic cell. Size of ribosome 70S in case of prokaryotic cell and 80S eukaryotic cell. Cell wall containing peptidoglycan case of prokaryotic cell. Difference between eukaryotic and prokaryotic cell is: in case of gram-positive, there is cell membrane, nucleate, flagellum, and cell wall. In case of gram-negative bacteria, there is pili, capsule, cell membrane, ribosome, cell wall, outer membrane, and granule. In case of eukaryotic cell, rough endoplasmic reticulum, nucleus, cytoplasm, and mitochondria. What is normal flora? Normal flora, they populate the healthy human body by the billions as benign passengers and as participants in bodily functions. Various bacteria and fungi that are permanent residents of certain body sites, especially skin, oropharynx, colon, and vagina. Viruses and parasites are usually not considered members of the normal flora, although they can be present in asymptomatic individuals. Beneficial functions of normal flora. The normal flora can provide some definite benefits to the host. First, they constitute a protective host defense mechanism. The non-pathogenic resident bacteria occupy attachment sites on the skin and mucosa that can interfere with colonization by pathogenic bacteria. The ability of members of the normal flora to limit the growth of pathogen is called colonization resistance. Secondly, some bacteria of the bowel produce antimicrobial substance to which the producers themselves are immune. Third, the bacterial colonization of the newborn infant acts as a powerful stimulus for the development of the immune system. Bacteria of the gut provide some important nutrients such as vitamin K and B vitamins and also aid in the digestion and absorption of nutrients. Harmful effect of normal flora. Clinical problem caused by the normal flora arise under the following conditions. When the organisms are displaced from their normal site in the body to an abnormal site. Example, normal skin bacteria Staphylococcus epidermidis into bloodstream where it can colonize catheters and heart valves resulting in bacterial endocarditis. When potential pathogen gain competitive advantage due to diminished population of harmless competitor. When normal bile flora is depleted by antibiotic therapy, leading to overgrowth by the resistant Clostridium difficile, which can cause a severe colitis. When some harmless, commonly ingested food substances are converted into carcinogenic derivatives by bacterial sulfatases of the sweeter cyclamate into bladder carcinogen cyclohexamine. When individuals are immunocompromised, normal flora can overgrow and become pathogenic. Pathogenesis. The pathogenesis of bacterial infection include initiation of the infectious process and the mechanisms that lead to the development of sign and symptom of disease. Outcomes following exposure to microorganism is colonization, organism eliminated by host defense, and infection. Infection organism become part of normal flora. Carrier state, no apparent symptom, but organism are shared. Pathogenicity causes infectious disease. What is pathogen? Few species of microorganisms that are harmful to humans either by production of toxic compounds or by direct infection are characterized as pathogen. Colonization. Establishment of proliferating microorganisms on the skin or mucous membrane. What is pathogenicity? Virulence factors, number of initial organisms, and immunostatus. What is virulence? Virulence is a quantitative measure of pathogenicity is measured by the number of organisms triggered to cause disease. The 50% lethal dose is the number of organisms needed to kill half the host. Infectious dose 50. The 50% infectious dose is the number of organisms needed to cause infection in half the host. Shigella infectious dose 50 is more than 100 organisms and Salmonella is more than 1000 organisms. Infectious dose depends upon their bacterial virulence factor. Villi allow the organism to adhere well to mucous membrane, whether they produce exotoxin or endotoxin. capsule to protect them from phagocytosis. What is parasite? A living organism, bacteria, virus, fungus, protozoa, and helminth, which receive nourishment and shelter from another organism where it lives. Obligate intracellular parasite. Some bacterial pathogens are called obligate intracellular parasite as they can grow only within host cells. Example Chlamydia and Rickettsia. Facultative parasite. Many bacteria can grow within the cell, outside the cells, or in bacteriologic media. Bacteria causes disease by two major mechanisms: toxin production, invasion, and inflammation. Type of bacterial infection: epidemic, outbreak, local, endemic, regional, and pandemic, widespread. Epidemic means which occurs more frequently than usual, and endemic means which present constantly at a low level in a specific population. Pandemic means if an infection has worldwide distribution. Stages of bacterial pathogenesis: transmission from external source into the portal of entry, evasion of the primary host defense such as skin or stomach acid, adherence to the mucous membrane usually by the bacterial pili, colonization of bacteria by growth at the site of adherence. Disease symptom caused by toxin production and invasion accompanied by inflammation. Host response both specific and non-specific during 3-4-5. That is adherence, colonization, and disease symptom. Progression and resolution of the disease. Determinants of bacterial pathogenesis, that is transmission, adherence to host cell surface, invasion, inflammation, intracellular survival, and toxin production and immunopathogenesis. Transmission means mode of transmission of bacteria and other infectious agents is most important because interrupting the chain of transmission is an excellent way to prevent infectious disease. Mode of transmission: human to human and non-human to human. Vertical transmission: a process of transmission of disease from mother to offspring across the placenta, neonatal sepsis, neonatal meningitis. Within birth canal during birth, neonatal meningitis and conjunctivitis, where the breast milk, oral or skin infection. Horizontal transmission: transmission of disease from person to person. There are four important portals of entry: respiratory tract, airborne droplet, GI tract, fecal contamination of food and water, genital tract, sexual contact, and skin contact. Human to human transmission: direct contact, gonorrhea, sexual herpes through birth canal, no direct contact, dysentery, fecal oral route, and transplacental congenital syphilis, bacteria cross the placenta and infect fetus. Non-human to human: soil entered, tetanus, spore in soil and in skin, water source, legionnaires disease, bacteria and water aerosol are inhaled into lungs. Animal source: directly cat stress disease enter in cat stress via insect vector, Lyme disease, bacteria enter by tick bite, via animal excreta, E. coli, hemolytic uremic syndrome, bacteria in cattle feces, ingested in undercooked hamburger. Adherence to the cell surface, certain bacteria have specialized structure, villi, capsule, and glycocalyx that allow them to adhere to the surface of the human cell, thereby enhancing their ability to cause disease. Villi contain example Neisseria gonorrhoeae and E. coli. Possess villi which mediate attachment to the organism to the urinary tract. Glycocalyx organisms Staphylococcus epidermidis and Pyridae streptococcus. The various molecules that mediate adherence to cell surface are called adhesins. Adhesion Bacterium adhesin epithelium. Foreign bodies such as artificial bulb and artificial joints predispose to infection. Bacteria can adhere to the surface, but phagocytes adhere poorly due to the absence of selectins and other binding proteins of the artificial surface. That protein, lipoteichoic acid, and fibronectin. Surface protein present in E. coli and Salmonella, which mediate binding of bacteria to endothelium and to extracellular protein, and also interact with serum protein factor 12 and play a role in the production of thrombi in sepsis associated with DIC. Invasion, inflammation, and intracellular survival. Several enzymes secreted by the invasive bacteria play a role in the pathogenesis. These are collagenase, hyaluronidase, coagulase, immunoglobulin A protease, and leukocidase. Collagenase and hyaluronidase, which degrade collagen and hyaluronic acid respectively, thereby allowing the bacteria to spread through subcutaneous tissue. They are especially important in cellulitis caused by Staphylococcus pyogenes. Ovulase produced by Staphylococcus aureus accelerates the formation of fibrin clot and from its precursor fibrinogen. Leukocidin, which destroys both neutrophilic leukocytes and macrophages. Immunoglobulin A protease produced chiefly by Neisseria gonorrhoeae, Haemophilus influenzae, and Streptococcus pneumoniae. Degrade the immunoglobulin A, allowing the organism to adhere to mucous membrane. Penetration and spread. Virulence factor contribute to invasiveness by limiting the ability of the host defense mechanism. Phagocytosis. Capsule antiphagocytic factor, surface M protein, antiphagocytic factor, protein A binds to immunoglobulin G and prevent activation of complement. Inflammation, pyogenic and granulomatous inflammation. Pyogenic gram-positive and gram-negative cocci, neutrophils are predominant. Granulomatous inflammation, Mycobacterium tuberculosis, macrophage and T cell are predominant. Intracellular bacteria cause granulomatous inflammation. Common bacteria are Mycobacterium, Legionella, Brucella, and Listeria. These organisms are not obligate intracellular like chlamydia and rickettsia, as they can cultured in microbiologic media in the laboratory. Different mechanisms allow them to survive and grow intracellularly. Inhibition of the fusion of phagosome with lysosome, which allows the organism to avoid the degradative enzyme in the lysosome. Inhibition of acidification of phagosome, which decreases the activity of the lysosomal degradative enzyme. Escape from phagosome into the cytoplasm where they are non-degradative enzyme. Intracellular parasite, no fusion, and enter cytoplasm. Exotoxins are produced by several gram-positive and gram-negative bacteria, in contrast to endotoxins, which are present only in gram-negative bacteria. Exotoxins are polypeptides secreted by the certain bacteria that alter specific cell function, resulting in the symptom of disease. Endotoxin is a component of the cell wall. Endotoxin are lipopolysaccharides located in the outer membrane only of gram-negative bacteria. They are not secreted by bacteria. Exotoxin and endotoxin. Main feature of exotoxin and endotoxin. Certain species of gram-positive and gram-negative bacteria. Endotoxin cell wall of gram-negative bacteria. Secreted from cell present. Exotoxin are polypeptide, endotoxin are lipopolysaccharide. Exotoxin plasmid or bacteriophage, endotoxin bacterial chromosome. Toxicity high in exotoxin. Clinical effect, various effect, and case of endotoxin fever shock. Mode of action various. In case of endotoxin include tumor necrosis factor and interleukin 1. Antigenicity induces high titer antibodies called antitoxin. In case of endotoxin, poorly antigen. Toxoid used as vaccine. In case of endotoxin, no toxoid found and no vaccine available. Heat stability restored rapidly at 60 degrees and stable at 100 degrees centigrade for 1 hour. Typical disease in case of exotoxin, tetanus, botulism, and diphtheria. Case of endotoxin, meningococcemia, sepsis by gram-negative drug. What is toxoid? When exotoxin treated with formaldehyde or acid or heat, the exotoxin polypeptide are converted into toxoid. Toxoid are used in protective vaccine because they retain their antigenicity but lost their toxicity. Secretion system. The exotoxins are released from bacteria by specialized structure. Some secretion systems transport the exotoxin into the extracellular space, but others transport the exotoxin directly into the mammalian cell. Those that transport the exotoxin directly into the mammalian cell are especially effective because the exotoxin is not exposed to antibodies in the extracellular space. Six types of secretion system. Type 3 injectosome is important in virulence, mediated by needle-like projection, molecular syringe, and by transport pump in the bacterial cell membrane. Secretion system. The medically important gram-negative dots that utilize injectosome, Pseudomonas aeruginosa, E. coli, Shigella, Salmonella, and Yersinia pestis. Mechanism of action of bacterial exotoxin. ADP ribosylation, diphtheria toxin, cholera toxin, E. coli head level toxin, and pertussis toxin. Superantigen. Toxic shock syndrome toxin, staphylococcal enterotoxin, and erythrogenic toxin. Mechanism of action protease, exotoxin, tetanus toxin, botulinum toxin, and scalp skin toxin. Lecithinase, Clostridium perfringens alpha toxin. Mode of action of diphtheria toxin. Active binding cell surface. Causes inhibition of protein synthesis. Mode of action of cholera toxin. Case of Bartholomew, Gas protein, adenylate cyclase, cyclic AMP, protein kinase, water and electrolyte, water, chloride and bicarbonate which causes diarrhea. Membrane damaging exotoxin, protease, phospholipase and detergent-like action. Superantigen. Toxin shock syndrome toxin, staphylococcal enterotoxin, and iatrogenic toxin. Superantigen mechanism of action helper T cell receptor, CD3 protein, superantigen. MHC class 2. T cell activation figure. What is endotoxin? Endotoxin are integral part of the cell wall of both gram-negative rods and cocci. The toxicity of endotoxin is low in comparison with that of exotoxin. All endotoxin produce the same generalized effect of fever and shock. Endotoxins are weakly antigenic. They induce protective antibodies so poorly that multiple episodes of toxicity can occur. No toxins have been produced from endotoxin. Endotoxin are not used as antigen in any available vaccine. Mode of action of endotoxin. Endotoxin is the most important cause of septic shock. The septic shock is characterized by fever, hypotension, and DIC. Endotoxin causes this effect by activating three critical processes. Endotoxin activate macrophage, activate complement, and activate Hazmat factor. Interleukin-1 causes fever. Tumor necrosis factor causes fever and hypertension. And nitric oxide causes hypotension. Activate complement C3A causes hypotension, edema. C5A neutrophil chemotaxis and activate Hazmat factor, coagulation cascade, and DIC. What is immunopathogenesis? In certain disease, the organism itself not causes the symptom of the disease, but immune response to the presence of the organism. Example, rheumatic fever and acute glomerulonephritis. In case of rheumatic fever, antibodies are formed against the protein of Streptococcus pyogenes, which cross-react with joint, heart, and brain tissue. Inflammation occurs, resulting in the arthritis, carditis, cholera that are characterized findings in this disease. Bacterial infections associated with cancer. The association of Helicobacter pylori infection with gastric carcinoma and the multiple lymphoma. The association of Campylobacter jejuni infection with lymphoma. Different strain of bacteria can produce different disease. Individual bacteria produce different virulence factors that endow those bacteria with the capability to cause different disease. The different virulence factors are encoded on plasmid, on transposons, on the genome, and on the pathogenicity island. This transferable extrachromosomal genetic element may or may not present in any single bacterium, which account for the ability to cause different disease. Non-pathogenic strain of E. coli, E. coli causing watery diarrhea, the enterotoxigenic strain, E. coli causing UTI, ureopathic strain, E. coli causing neonatal meningitis, SCAN1, capsular strain. Typical stages of infectious disease. Incubation period, which is the time between acquisition of the organism or toxin and the beginning of symptoms. The time varies from hours to days to weeks depending on the organism. What is prodromal period, which occur non-specific symptoms such as fever, malaise, and loss of appetite occur. The specific and illness period, during which the overt characteristic sign and symptom of the disease occur. The recovery period, which occurs the illness abates and the patient returns to the healthy state. After recovery period, some individuals become chronic carriers of the organisms and may shed them while remaining clinically well. Some may develop a latent infection, subclinical infection. The individual remains asymptomatic although infected with the organism. Which is the pathogen? If a particular microorganism is isolated from an infected tissue, for example, from a necrotic skin lesion, one cannot conclude that the isolated organism caused the lesion. The organism could be a member of normal natural skin flora. Alternatively, the organisms may not be a natural resource of the skin, but an opportunistic pathogen that secondarily infected the necrotic lesion. In 19th century German microbiologist Robert Koch faced this problem when he was trying to determine the cause of an infectious disease. He defined a series of criteria, Koch's postulate, by which the identity of the causative microbial agent of a disease can be confirmed. Koch's postulate, etiological relationship between an organism and a particular disease. He identified a microorganism, cultured the microorganism out of the body, and injected it into the animal. The animal then got the disease. These criteria are: the microorganism must be isolated from every patient with the disease, but not in healthy ones. The microorganism must be isolated free from all the organism and grown in pure culture in vitro. The isolated microorganism must cause the original disease when inoculated into a susceptible animal. The microorganism must be re-isolated from the inoculated animal. Demonstration of Koch's postulate. How bacteria evade the host immune response? Pathogenic bacteria evolve mechanisms that facilitate their survival as successful parasites. Some bacteria evade host immunity by avoiding detection. They may express non-immunogenic determinants, may shed, modify, or alter their antigen, may interfere with antigen processing, or may even mimic or assume host antigen. Some bacteria produce inhibiting product that help them to survive, as for example, Staph aureus secrete leukocidin, a toxin lethal for neutrophils. Pseudomonas aeruginosa exoprotease may clip and hence inactivate immunoglobulin. Some Shigella species kill phagocytes after their ingestion. Some bacteria use several different mechanisms to allow them to survive and grow intracellularly and protected them from antibody and neutrophils that function extracellularly. This is all about normal flora and bacterial pathogenesis. Laboratory diagnosis of bacterial infection. Two main approaches. Bacteriologic organism is identified by staining and culturing the organism. And immunologic, which are serologic organism, is identified by detection of antibodies against the organism in the patient's serum. Bacteriologic approach. Specimen collection, stain the specimen, culture the specimen, identify the organism using appropriate test and perform antibiotic susceptibility test. Bacteriologic approach: specimen collection, choose appropriate specimen, obtaining the specimen properly, and transporting the specimen properly. Stain the specimen, gram stain or acid fast stain. Culture the specimen on the appropriate media. Identify the organism using appropriate test, that is hemolysis, pigment production, sugar fermentation, DNA probes, agglutination test, and immunofluorescence. Perform antibiotic susceptibility test. The figure of Ramstein. The figure of acid fast bacilli is red, rod, and beaded appearance. Figure of catalase test. Test tube contain hydrogen peroxide. Catalase positive, produce bubble. Catalase negative, no bubble. Positive bacteria are Staphylococcus and Enterobacteriaceae. Staggered plate of McConkey's agar media. The blood agar media showing hemolysis. It is golden yellow color. Properties. Mannitol salt agar media. Inoculating wild loop for the subculture touch a single colony and inoculate in triple sugar iron. Triple sugar iron agar stain. Fermentation of glucose, lactose or sucrose. Reduction of sulfur to hydrogen sulfide. This causes gas formation used for enterobacteriaceae. Oxidase negative. This is oxidase test. A drop of oxidase reagent. Oxidase reagent is added on filter paper. Bacterial colony is smeared on the paper. In positive case, purple blue develops in a few seconds. Negative case, no color develops. Purple color. Oxidase positive. Pseudomonas vibrio mesorio. No purple color. Antibiotic susceptibility test. Determination of minimal inhibitory concentration and the disk diffusion test. Immunologic method: using known antibody to identify the microorganism and using known antigen to antibodies in the patient's serum. Immunologic detection. Instant view test. Capsular swelling reaction, antisera against Staphylococcus pneumoniae, Haemophilus influenzae, and Listeria meningitis. Slide agglutination test, antisera against Salmonella and Shigella. Latex agglutination test, latex beads coated with specific antibody. Counter immunoelectrophoresis test, enzyme linked immunosorbent assay, and fluorescent antibody test. Slider tube agglutination test, typhoid fever, brucellosis, plague, leptospirosis, and facial disease. Serological test. Indication of blood culture: sepsis, endocarditis, osteomyelitis, meningitis, and pneumonia. Important pathogen present in blood culture: Staphylococcus aureus, Streptococcus pneumoniae, Escherichia coli, Klebsiella pneumoniae, and Pseudomonas aeruginosa. 10 ml blood at least 3 samples in 24 hours. Must be clean with 2% iodine to prevent contamination by the member of the flora of the skin. When heart infection, checked for turbidity daily for 7 days or longer. If both occur, Ramstein subculture and antibiotic sensitivity test. This is all about normal flora, pathogenesis of bacteria, and the laboratory diagnosis of bacterial infection.